అమృత. ఓకే. ఓన్లీ 19 మెంబర్సే ఉన్నారమ్మా, చూడండి ఒకసారి. ఏం చేస్తూ ఉంటారో ఏం అర్థం కాదు. ఒకసారి ఏమైనా జాయిన్ అవ్వాల్సిన వాళ్ళు ఉంటే ఒకసారి కాల్ చేయండి అమ్మా. దెన్ ఐ విల్ స్టార్ట్ ది క్లాస్.
ఓకే సార్.
ఒక్కసారి కాల్ చేయండి, రిమైండ్ చేయండి వాళ్ళు. Okay, so have you contacted them? So only one person was joined. Emma. See again, I am going to read the attendance here. Who is present here? Jyotirmay is present. Satvika is present. Sunita present. Guru Shankar, Suresh, Sandhya Rani, Lokeshwari, Obalesh, Sumakirtana, Muni Bhargavi, Pavani, Ashok, Raghuvamsi, Rajesh, Vineet, Bashir, Farhana, Jitendra Satyakrishna, Srinitya, and Vasavi Sheshavalli. Right, so I think one more is added now. So have you called anybody? కాల్ చేశారా? రెడ్ అయ్యా వీళ్ళకి చేయండి ఒకసారి. ఏం చేస్తా ఉన్నారు వీళ్ళంతా? నిద్రపోతున్నారు తినేసి. Okay, so let me have your attendance then. So I'll ask her.
Sir, again we have class at 3. Start it, sir.
You have class at 3, but I don't see all of your classmates. What can I say? What can I do? వాసు ఫస్ట్ ఇయర్ వాళ్ళ లిస్ట్ తీసుకురా ఒకసారి ఇట్లా. Yeah. No, what can I do? See, because if they miss and then I can't explain it again and again. Okay, so anyhow, I'll start this class. Okay. I still 9 more members need to be joined. I don't know what they are doing. Huh? Is anybody called anyone? You just tell me.
మెసేజ్ చేశాను సార్.
మెసేజ్ చేస్తే వాళ్ళు చూసుకోవద్దయ్యా, నేను ఆల్రెడీ పెట్టా కదా క్లాస్ అని. నేను ఆల్రెడీ క్లాస్ అని పెట్టా కదమ్మా. ఫోన్ ఉంటే కాల్ చేసి చెప్పండి. దట్ మే బి ద బెటర్ వే. Okay, so anyhow I'm going to start this. So in the yesterday class and in the previous classes, we have seen the various aspects which are related to the molecular biology, right? So we studied about central dogma of molecular biology, and then we were also seeing what is the importance of nucleic acids, how they form, and then what is the difference between RNA and DNA and so on. That we have discussed in the last classes. And now you can see the DNA replication when we come back to the, because I told you the molecular biology is having four kinds of aspects which I are going to study, and the first one is like the first unit always deals about the DNA replication. That means we are following according to the central dogma. So the first topic or the first unit is representing the replication of DNA molecule, and the second unit is a transcription of the RNA molecule, and the third unit deals about the expression and the genetic code, so which is a base for the translation process. So the translation is the fourth unit where we study about the ribosomes and all, and then we'll go for the protein synthesis and its what we can say the mechanism of action both in prokaryotic and eukaryotic system. So that we will see, right? So in that case, what happens, until yesterday we completed the basics of DNA replication. So the basics of DNA replication always helps to identify or like basics of molecular biology, right? So Nagarazu is added. Okay. Yeah. Okay, so the basics of molecular biology, so we have discussed, and which helps us to lead into the main topic. So the first lead main topic is like DNA replication. So in the DNA replication, what we have, what we'll see, so the DNA molecule will get duplicated. So the duplication of DNA molecule always will help, so helps to what I can say like doubling of DNA molecule. So usually in the cell cycle, what I explained in the previous slides, in the previous classes, so there you understood that the cell cycle is always having a phase called S phase. S phase can also be called a synthetic phase. In the synthetic phase, what will happen? A large amount of DNA molecule, or you can say that the duplication of DNA molecule will take place. The duplication of DNA molecule is always takes up because of DNA replication. So the DNA replication, in the sense, we'll have like two strands from the, one is from the father and the other one is from the mother in case of chromosomes. And in that you can see that the double stranded DNA molecule, the double stranded DNA molecule will undergoes like replication process where it produces, instead of one double stranded DNA, you can see that there are two double stranded DNA molecules, right? That's what we can see. Okay, so that is nothing but the replication process, what we call. And the replication process, before going to that, you should also know what is the importance of nucleic acids. So nucleic acids will act as genetic materials, and the genetic material always helps to have, so to transfer or to carry the characteristic properties from one generation to the other generation. So that's why we can call this as hereditary material. And mostly the nucleic acids are of two types. You can say that the DNA, of course, almost all eukaryotic organisms are holding the DNA as a genetic material. And of course, not only eukaryotes, you can also see that the many of the what I can say like prokaryotic organisms are also having DNA as a genetic material. RNA is also a genetic material in some prokaryotic species, which you can specifically call them as like the viruses, some viruses. So these viruses are having RNA as a genetic material. Especially the adeno type of viruses are holding the RNA as a genetic material. So that's what only the deviation which we can see with these nucleic acids. Then the cells of living organisms, they possess the same quality and quantity of DNA. You can see that the same quality and same quantity of DNA. That's what I already told you in the previous classes. If you take any cell in the organism, if you take a hair cell, 23 pairs of chromosome. If you take a nail, you can have a 23 pairs of chromosome. So you can take any cell in case of humans. So each cell will have the same quality and quantity of DNA. Same quality in the sense, so the purity of DNA is same, and the quantity is nothing but 23 pairs of chromosome. But I told you, not all the genes or not all the portions of the DNA can get expressed in the all the cells, right? So that's why we have a division of labor in the cells. Division of the labor is always a helps to, what is that, what will happen with this, a division of labor in the sense, so the cells will be divided and these cells will have a different kind of functions, like uh, when they divided, so there they can have, like, these group of cells will have this function, these group of cells will have this function, something like that. They will be divided at the time of what we can say, like, differentiation. So the differentiation will takes up, so that different systems will form. While forming the systems, and then they'll have their own special kind of characteristic properties, and then the genes, some of the genes will express only in that particular tissue. The best example here is the pancreas. So the pancreas can produce insulin gene, insulin protein, which controls the glucose. So the similar way, what happens, that means insulin gene is present in all the cells, but you can see only insulin cells we can produce the insulin molecule, right? So it's a qualitatively, the DNA molecule in all the cells of an organism is same, and the quantitatively also it is the same, right? And the DNA replication can be happen in two ways. One is called as like DNA directed DNA synthesis, or you can also call it as like RNA directed RNA synthesis can also be there. So where the DNA molecule will act as a template molecule, and so that the DNA polymerase can synthesize the DNA molecule. So that is called as synthesis of DNA molecule from DNA as a template. So the DNA will be act as template, and then the new strand will be synthesized, that is called as new DNA molecule. So in this case, this type of synthesis can be called as DNA directed DNA synthesis. Then you can also see RNA directed DNA synthesis. That means we know that the RNA is the genetic material in some of the prokaryotic systems, right? So that's what I told you here, right? That's it here, right? So when there are RNA viruses, RNA as a genetic material, then when it enters into the eukaryotic cells, so that means viral particles, they doesn't have any replication process outside the biological system. So when they attack the human cells, when they attack the some host cell, then automatically RNA directed DNA synthesizing system containing uh, viral particles, what happens, they will have a special system where they can produce the enzyme called as reverse transcriptase. So the reverse transcriptase is an enzyme which converts the RNA molecule into DNA. Okay, I see. Wait a minute, ma. అందరు రాలేదు వాసు. ఏం చేస్తున్నారు? 24 మంది ఉన్నారు. ఒకసారి చెప్తా, నోట్ చేసుకో. రాని వాళ్ళకి ఒకసారి కాల్ చేయి. Yeah, the attendance is like Rupa Jyotirmayi, Guru Reddaya, Satvika,
Sir.
okay, Sunita, Guru Shankar, Kavya, Suresh, then Sandhya Rani, Lokeshwari, Oblesh, Sumakirtana, Pavani, Ashok, Raghuvanshi, Rajesh, Vineet, Rajesh, Vineet, చెప్తా చూడు. కావ్య వచ్చిందా? సురేష్ వచ్చి, సంధ్యారాణి, లోకేశ్వరి, ఓబులేష్, సుమకీర్తన, ఫర్హానా, జితేంద్ర, సత్యకృష్ణ, శ్రీనిత్య, కళ్యాణి, శేషవల్లి. 24 మెంబర్స్ వచ్చారు. 24 వచ్చారు. మిగిలిన వాళ్ళ సంగతి ఏం చూడాడా? కాల్ చేయాలి అందరికి. Okay, so the RNA directed DNA synthesis can happen in case of viruses, where the RNA molecule enters into the host system, then it controls the host regulatory metabolisms, and then it produces a special kind of enzyme called as reverse transcriptase. A reverse transcriptase is the enzyme which synthesizes the RNA molecule into DNA molecule. So it synthesizes the DNA molecule, but the DNA molecule is synthesizing from RNA molecule. So that's what we can see in the RNA directed DNA synthesis. Okay, we'll see one by one in the next classes also. Then the general features of DNA replication when we talk about, so it has like, there are three theories developed to explain the DNA replication process. So in general, the replication process is explained in three ways, which we can call it as like conservative mode of replication, and dispersive mode of replication, and semi-conservative mode of replication. Remember all these three types are called as like hypothetic theories. They are hypothetic theories which explains about the replication process in the DNA molecule. Are you clear? Is it clear?
Yes, sir. Yes, sir.
Okay.
Yes, sir. Clear, sir.
Okay, so there are general features like the hypothesis అంటే just predictions అన్నమాట. అంటే మనకి మీకు మ్యాథమెటిక్స్ లో గుర్తుందా, యూ హ్యావ్ లైక్ పైథాగరస్ థియరమ్ ఆర్ సంథింగ్ లైక్ దట్ థియరమ్స్. Do you remember?
Yes, sir.
So when we have the theorems are always like it's a kind of like predictions. We are going to have a prediction. It may or may not be true. So if it is true, and then it will come into the force. Otherwise, it won't be come into the force. Okay. So similarly, the what happened? So to explain the DNA replication, there are three theoretical methods were proposed or developed for the double standard DNA replication process by various scientists, and they are nothing but conservative mode of replication, dispersive mode of replication, and then semi-conservative mode of replication. And let's see one by one. So the first one is a conservative mode of replication. So the conservative mode of replication is proposed by a scientist called Crick. Okay, so in this one you can see that the one daughter cell will have two new DNA strands, and one is having only the two parental DNA strands. Do you understand this? Are you clear?
Yes, sir.
If it is yes, then tell me what is happening here. One daughter cell synthesizes two new DNA strands, but whereas another one will have all two parental DNA strands. Two parental DNA strands are in the second one. Then how it is possible? You just imagine.
Mitosis, meiosis like division ఉంటుంది sir.
Mitosis meiosis will be happen only after the DNA replication process. You think logically, when the parental strand is there and the parental strand is going straight away to the one of the cell, one of the two daughter cells, and they says that the new strand will be generated by a new cell. Is it possible? I want to answer.
Okay, sir.
Is it possible? మీకు అర్థమవుతుందా నేను చెప్పింది? So in the picture you have one parental strand, right? And this one parental strand is going to the one daughter cell. and the other daughter cell will synthesize the completely two new strands, right? ఇదన్నా అర్థమైందా లేదా?
చెప్పండి సార్.
నేను చెప్పేది కాదమ్మా, మీకు అర్థమైందా లేదా? మీకు అర్థం కాకపోతే నేను చెప్పాలా? అర్థమైంది. Then tell me, how come that if the parent two strands will go into the one cell, then how come that the other cell will synthesize the new strands? What is the basic rule in the synthesis of DNA molecule? It requires template strand, is it not? మీకు అర్థమైందా అర్థం కాలేదా? మా అర్థం కాకపోతే చెప్పండి. If you are not understood this, and then I may not go further. చెప్పండి.
మళ్ళీ చెప్పండి సార్.
మళ్ళీ చెప్పాలా? ఓకే. యాజ్ పర్ ద కన్జర్వేటివ్ మోడ్, కన్జర్వేషన్ అంటే ఏంటి? ఇట్స్ నథింగ్ బట్ సేవింగ్, రైట్? కన్జర్వ్ అంటే ప్రిజర్వ్ చేయడం, రైట్? సో ఐల్ గివ్ యూ సమ్ కన్జర్వేషన్ మెథడ్స్ లైక్ conservation of germ cells, conservation of what I can say like blastocysts, conservation of stem cells. అంటే క్రయో బ్యాంక్స్ ఉన్నాయి తెలుసు కదా. ఇప్పుడు రీసెంట్గా ఏంటంటే పిల్లలు పుట్టారనుకోండి, వాళ్ళ కార్డ్ బ్లడ్ సెల్ నుంచి, అంటే మనం బొడ్డు అంటాం కదా, మదర్ నుంచి, ఆ కార్డ్ బ్లడ్ సెల్స్ ని సేవ్ చేస్తారు తెలుసా?
Yes, sir.
So those cord blood cells are nothing but stem cells. Stem cells are pluripotent and totipotent cells. I don't say it is totipotent, they are pluripotent cells. You know what is totipotent and then pluripotent, right? తెలుసా తెలీదా? తెలియకపోతే తెలియదని చెప్పండి, ఏమో నష్టమేం లేదు, తప్పేం కాదు. టోటిపోటెన్సీ అంటే ఏంటి? బాటనీలో చదువుంటారు కదా సార్, టిష్యూ కల్చర్లో.
కొత్త ఆర్గానిజం ని డెవలప్ చేసేదాన్ని టోటిపోటెన్సీ అంటారు సార్.
A new organism will develop, or like sure ఒక్కసారి రిఫైన్ చేసుకోండి దాన్ని. ఎనీబడి? ఆ టోటిపోటెన్సీ అంటే ఏంటి? Okay. See, totipotency is nothing but developing a, or you can say the capacity of a single cell, in the capacity of a single cell to develop into a complete organism. What do you mean by the complete organism? Especially the tissue culture, what happens, the plant cells will be taken, so they will be made into complete plant. So that means we are going to take only single cell, that single cell it will become into complete plant. That is called as totipotency. అంటే ఒక సెల్ కి, ఒక సెల్ దాని విలువ అంటే if that cell can able to produce a complete plant, that capacity is called as totipotency. అర్థమైందా?
Yes, sir.
So
Okay, sir.
totipotency is like developing a single cell into a complete organism is called as totipotency. Adi, that will be possible only in the plants but not in the animals. Okay, so in the animals what will happen? Instead of totipotency, you can see that the stem cells are pluripotent. So the pluripotent in the sense they can be developed into a particular organ, right? The cells can divide and then they form as organ. మనకి గుర్తే ఉండుంటుంది రీసెంట్ గా మనకి ఆర్టిఫిషియల్ హార్ట్ ఫిక్స్ చేస్తున్నారు, ఆర్టిఫిషియల్ లంగ్స్, ఆర్టిఫిషియల్ కిడ్నీస్, ఇట్లా చాలా వచ్చేసినాయి. ఆర్టిఫిషియల్ అంటే they are not like complete artificial, so they were made like this with the hypothesis they were made in the outside, so that you can see. Okay, so the conservation in the sense the conservation is always like saving. So మనకి క్రయో బ్యాంక్స్ ఉన్నాయి కదా, సో ఇన్ ద క్రయో బ్యాంక్స్ యూజువల్గా ఏం చేస్తారు, ఏం ప్రిజర్వ్ చేస్తారు? అసలు వినలేదా క్రయో బ్యాంక్ అనే మాటే మీరు? అడిగేది మిమ్మల్ని అమ్మా.
No, sir.
వాటిని ప్రిజర్వ్ చేస్తారు సార్.
Yeah, so any kind of cells can be preserved in the cryopreservation, right? So those cells may be used for further usage. అంటే యూజువల్గా ఏం చేస్తారంటే, let me give you an example here. For example, there are like the bulls, ఎద్దులు అంటాం కదా, రైట్? సో ఇప్పుడు మనకి ఎద్దులు అవన్నీ ఆల్మోస్ట్ చచ్చిపోయినాయి, ఎక్కడ లేవు. అంటే ఎక్కడో ఉన్నాయి కొన్ని, బట్ నాట్ దట్ హై నెంబర్. సో ఏం చేస్తారంటే వాటి ఆవు నుంచి ఎగ్ తీసుకొని అట్లాగే బుల్ నుంచి స్పర్మ్ తీసి అవన్నీ ప్రిజర్వ్ చేస్తారు. సో దట్ ఈస్ కాల్డ్ ఎస్ క్రయో ప్రిజర్వేషన్. సో దే విల్ బి అలైవ్ వెన్ ఎవర్ యూ టేక్ దెమ్ అవుట్. సో దెన్ if you go for fertilization between the egg and then sperm, and then you can get the bull or whatever it is, like the same species, okay, male or female animal. ఇది అర్థమవుతుందా? ఓకే, క్రయో ప్రిజర్వేషన్ అంటే క్రయో ప్రిజర్వేషన్ కెన్ ఆల్సో బి డన్ ఫర్ లైక్ ఎండమిక్ ప్లాంట్స్, ఎండమిక్ యానిమల్స్, వేటికైనా ఉపయోగిస్తారు, రైట్? Okay, so the conservative mode explains that completely the one parental DNA, that means the parental DNA molecules are enters into the one daughter cell. That means the other daughter cell is not having any DNA molecule, but we don't know how come the second daughter strand is synthesized here, because when the other strand is need to be synthesized, then automatically it requires a template strand. So it is already there, right? When it is already there in the parents, then from there what will happen? The other strands will get synthesized. But you see, as per the conservative mode of replication is like it is conserved to only one cell. Parental DNA is conserved to only one cell. So ఒక సెల్ లోకి రెండు DNA మాలిక్యూల్ వెళ్ళిపోతే అండ్ సెకండ్ సెల్లో DNA మాలిక్యూల్ ఎలా వస్తుంది? బికాజ్ we need a DNA strand for synthesizing the other DNA strand. What we call it as like a template strand. So the template strand is always required to synthesize the next strand. So then how come in the conservative mode of replication, the second cell, the first cell is having the parental DNA and the second cell is not having? How come that second cell can synthesize the double stranded DNA without any DNA molecule? So this is my question. Is it true or not? I want answer.
Sir, ask once again the question, sir.
Once again the question. Okay, so one cell is completely having DNA from the parents. That means the two sets of parental chromosomes will come into the one cell. That cell is safe, but we have a second cell. So the second cell is going to produce the DNA molecule. So how come it produces? Is it produces or is it not produces? Because DNA replication always requires a template molecule. If there is no template molecule here, then how come that it will get synthesized? the double stranded DNA molecule. ఎందుకంటే డిఎన్ఏ సింథసైజ్ అవ్వాలంటే ఏదో ఒక బేస్ ఉండాలి కదా, ఒక స్ట్రక్చర్. దట్ స్ట్రక్చర్ ఈజ్ నాట్ హియర్ ఇన్ ద కన్జర్వేటివ్ మోడల్. ఇది క్లియరేనా? Okay, let me tell you here one thing. You see that there are two strands here, right? The parental strands. So these two parental strands will get separated. Then each strand will synthesize the new strands. So that means this strand will involve and then it synthesizes another one, and this one involve and then it synthesizes another one. Are you clear up to now? ఇక్కడ అర్థమైందా?
సార్ పీపీటీ కనిపించడం లేదు సార్.
సార్ మీరు స్లైడ్స్ మూవ్ చేస్తే
యా ఓకే సార్, క్యారీ ఆన్.
పీపీటీ కనబడట్లేదా మీకు? ఎందుకు కనపడదు?
ఫస్ట్ కనిపించలేదు
లేదు సార్, అంతేపటి నుంచి కనిపించలేదు సార్, ఇప్పుడు కనిపిస్తుంది.
Okay, so let's see this strand. You see this strand, parental strand, right? So in the parental strand, this strand is involved in synthesizing another strand. కొత్త స్టాండ్ వస్తుంది దీనికి, అలాగే దీనికి కూడా ఇంకొక స్టాండ్ వస్తుంది, రైట్? టూ స్టాండ్స్ లో ఒక స్టాండ్ ఒక స్టాండ్ న్యూ స్టాండ్ ని ఇంకొక న్యూ స్టాండ్ ని సింథసైజ్ చేస్తాయి. అర్థమైందా ఇది? ఆర్ యూ క్లియర్?
యా ఎస్.
So parents లో there are two strands, and out of two strands, one strand is involved in one new strand synthesis, another strand is involved in the another new strand synthesis. Okay, but in case of conservative model, what is happening? The total DNA which is here, you see this DNA, this double stranded DNA is going straight away into the daughter cell, and the other daughter cell doesn't have any DNA to synthesize, but they says that Crick says that it also synthesizes the new strands. That's it. How it is possible? It is not possible because by the time when they proposed, so during that time they doesn't know clearly about the replication process, how it happens. So that's why they have proposal like this. Do you understand what I mean? So the conserved ante, so parental DNA as such, it is going to the one daughter strand, that is conserved DNA, right? It is conserving, saving from parent to daughter cell completely. It's not like a partial conservation, right? ఇది అర్థమైందా?
యా ఎస్ సార్.
I want answer from others.
సార్
Sure? I don't know, it's not so easy to understand because it is difficult. So when it is on the blackboard, then I can write something. But whereas here, so I can't help you. Okay, so one parental strand or the parental strand as such, it enters into the one daughter cell, and then other cell will synthesize the new strand. And similarly, these two cells will also undergo another next division. So when they go next division, then this whole DNA molecule enters here, right? And then similarly, the whole DNA molecule enters into here, and this one are the new cells, and they will synthesize again the new strands. So this is what they said, like say a conservative mode of replication. So it is conserved into a one daughter cell, and likewise several daughter cells will be continued from one to other. So when there is a cell division, are you clear with this? అర్థమైందా?
ఎస్ సార్.
So in the conservative mode, so one daughter cell will highly having the, or it is completely the genetic information from the parents will be transferred to the one cell, and the other cell will synthesize the new strand. So similarly, you can see that each cell will have like that, each division will have like that, and finally you can see first replication, second replication, third replication, and so on. And you can see that the parents are somewhat distant from that particular species, like grandparents or something like that. So in the conservative mode of replication, the parental DNA, two strands of parental DNA enters into the one daughter cell, the other daughter cell will synthesize the new strand, and the similar way in the second generation you can also see this blue color one. This one is the one which is synthesized in the first generation, and it carry forwards to the second generation, right? And the new cell will have the new strand, and similarly here also you can see the new strand and then highly conserved strand. Okay, that is called as conservative mode of replication. The next is dispersive mode of replication. So in the dispersive mode of replication, what will happen? There are two strands in the parental DNA, and two strands will enter into one strand will enter into one daughter strand, daughter cell, and the other strand will enters into the other daughter cell, right? Then what will happen? So these strands, the parental strands, one strand and one strand, these strands maybe will act as a complementary DNA molecule or the template DNA molecules to synthesize the next strand. So when the next strand will get synthesized, then automatically the strand which is coming from the parent will get disappear, degraded. Okay, again what will happen? So the newly synthesized strand will become the single stranded one, or you can say like newly become one can go for another cycle. So that means what will happen? The double stranded DNA molecule will form. Do you understand? So in the middle of the reaction, what is happening? There is a disappearance of the DNA molecule. So that's why dispersion of DNA molecule. That's why this method is called as dispersive model of DNA replication. Is it clear? ఎవరికి అర్థం కాలేదా?
సార్ అదొకసారి స్లైడ్ చూపించి చెప్పరా సార్?
స్లైడ్ కనపడట్లేదా మీకు?
కన్జర్వేటివ్ మోడల్లో కనిపిస్తుంది సార్.
మార్చాను కదమ్మా. యు డోంట్ సీ స్లైడ్స్? చూడలేదా? లేదా?
కన్జర్వేటివ్ మోడల్ కనిపిస్తుంది సార్.
ఇప్పుడు కనిపిస్తుందా?
ఎస్ సార్.
వై ఇట్ ఈస్ హ్యాపెనింగ్? నేను పవర్ పాయింట్కి వెళ్ళినా కానీ కనపడలేదంటే మీకు ఎట్లా? ఇప్పుడు ఫుల్ స్క్రీన్ కనిపిస్తుందా మీకు?
కనబడుతుంది.
Okay. Okay. So you see here, this is a parent strand. When it is a, we can say like conservative model, the parent strand will go into the one daughter cell and the new strands will synthesize in the another daughter cell. Whereas I'm not going to talk about semi-conservative, I'm going to talk about the dispersive method. So in the dispersive method, you see that there are two strands here, right? Is it not?
Yes, sir.
So this strands, one strand will enters into the one daughter cell, another strand will enters into the another daughter cell. Then automatically, because they are single stranded, so they can synthesize the new strand. So this is called as dispersive method. Actually ఏం జరుగుతుందంటే ఇక్కడ DNA molecule will get dispersed, డిగ్రేడ్ అయిపోతుంది, అందుకనే డిస్పర్సివ్ మెథడ్ అంటారు, రైట్?
ఎస్ సార్.
ఏం రైట్ ఏమో అసలు మీకు ఏం అర్థం అవుతుందో నాకేం అర్థం అవట్లేదు. ఓకే. సో యాక్చువల్గా డిస్పర్సివ్ మెథడ్లో ఏంటంటే ఇది తప్పు. ఈ పిక్చర్ యాక్చువల్గా ఈ పిక్చర్ తప్పు. ఇక్కడ 2 రెడ్ కలర్స్ ఉన్నాయి కదా, ఆరెంజ్ కలరు, సారీ ఇది అది పింక్ కలర్ స్టాండ్స్, రైట్? ఇక్కడ సింథసైజ్ అయిన వాటిలో ఒక దానిలో పింక్ కలర్, ఒక దానిలో వేరే కలర్ ఉండాలి. సో దాట్ ఈస్ రైట్ పిక్చర్. ఇది కాదు. ఇది అర్థమవుతుందా? I don't know.
ఎస్ సార్, అర్థమైంది సార్.
So there are 2 strands in the parents, and one strand will go to one daughter strand and the other strand will go to other daughter strand, and they will be involved in the synthesis of a new strand. So both the cells are having one parental strand, one is from a newly synthesized strand, right? That's what you can see. And even in the second generation the same thing will happen, right? So out of these 2 strands, one strand will go into one daughter cell, other one will go to other daughter strand. So the same is the case, right? Are you clear?
Yes, sir.
ఎస్ సార్.
Okay, so the dispersive mode of replication is also not so good, and it was difficult to explain what will happen in the complex systems. So that's why the dispersive model is also not having any experimental proof. So what happened? This is no more using by the scientists. So we are not going to use this. The next is semi-conservative mode of replication. So semi-conservative mode of replication, what will happen? So you can see double strand DNA synthesis. Then how it happens that things. Slide కనబడట్లేదా? ఏందమ్మా? You see now?
ఎస్ సార్, ఇప్పుడు కనబడుతుంది.
ఇప్పుడు మీకు ఫుల్ స్క్రీన్ వచ్చిందా?
ఎస్ సార్.
ఓకే. సో నేను సెమీ కన్జర్వేటివ్ మోడ్ రేపు చెప్తాను మీకు. ఎందుకంటే ఐ డోంట్ అండర్స్టాండ్ దట్. మీరు ఐఎమ్ నాట్ కంఫర్టబుల్. మీరందరూ డిస్పర్సివ్ తర్వాత కన్జర్వేటివ్ మోడ్ అర్థమైందంటే నాకు అర్థం అవ్వట్లేదు. ఐ డోంట్ నో ఏంటి
అర్థమైంది సార్.
ఏం అర్థమైందయ్యా? అంతా ఎక్కడో ఉన్నారు, ఈ లోకంలో లేరు. Because if you don't understand this and then further class you may not understand. That's my concern. Okay, maybe I'll try to explain you in the whiteboard. Let me check whiteboard here. ఈ వైట్ బోర్డ్ కనిపిస్తోందా?
బ్లాక్గా ఉంది.
వస్తుందమ్మా, వస్తుంది.
Yes, sir.
వచ్చిందా వైట్ బోర్డు?
Yes, sir.
ఓకే, గ్రేట్. సో
సార్ నాకు కనిపించలేదు సార్ బియ్యం బోర్డు.
నాగరాజు కనపడలేదా?
కనపడలేదు సార్.
వైట్ బోర్డ్ అంటే ఇందాక షేర్ అయిన దగ్గరనే నీకు జస్ట్ తెల్లగా వస్తుందమ్మా, అంతే.
తెల్లగా ఏం రాలేదు సార్, బ్లాక్ గా ఉన్నాయి బాక్స్ లో.
బ్లాక్ గా ఉన్నాయి
సార్ మళ్ళీ బ్యాక్ వచ్చేసింది సార్.
అవునమ్మా, అవును, ఐఎమ్ డూయింగ్ ఇట్. మీరు ఎవరు ఆపరేట్ చేయొద్దండి, జస్ట్ కీప్ క్వైట్. వైట్ బోర్డ్లో అయితే నేను జస్ట్ డ్రా చేసి చూపించవచ్చు అందుకోసం అని మీకు. ఇప్పుడు వచ్చిందా?
ఎస్ సార్. సార్
అందరికీ వచ్చిందా? నాగరాజు వచ్చిందమ్మా?
నో సార్, రాలేదు.
రాలేదా?
రాలేదు సార్.
వైట్ బోర్డ్ అంటే తెల్లగా ఉంటుంది అంతే. దాని మీద స్కెచ్లు కనిపిస్తున్నాయా?
ఏం కనిపించలేదు సార్.
ఏమి
ఒక
కనిపించలేదు.
నేర్పించాను అంటే.
ఓకే, మేబీ మీకు అది సరిపోవట్లేదమ్మా, సరిపోనట్లేదు. ఓకే, దెన్ నేను ఐ విల్ ఎక్స్ప్లెయిన్ యూ టుమారో. ఓకే, సో ఎనీహౌ ఇట్ ఈస్ ఆల్మోస్ట్ 3 o'clock నౌ.
ఓకే సార్.
యా, మళ్ళీ రేపు ఇంకొకసారి మళ్ళీ స్టార్ట్ చేసి చెప్తానులేండి కంప్లీట్గా. ఓకే, కన్జర్వేటివ్, సెమీ కన్జర్వేటివ్ అండ్ దెన్ కన్జర్వేటివ్, డిస్పర్సివ్ అండ్ సెమీ కన్జర్వేటివ్ మోడ్స్. ఓకే
ఓకే సార్.
ఓకే, ఇక్కడ ఎవరు ఇంకా రాలేదు? మొన్నటి వరకు అందరు వచ్చారు, మళ్ళీ ఇప్పుడు రావట్లేదు ఏంటి? See, those who are not having, the access, they may not be having this thing. మేబీ మీరు డిఫరెంట్గా చేసుకున్నట్టున్నారు. మా నాగరాజు, బషీర్ వీళ్ళందరూ గెస్ట్గా వస్తున్నారు కదా బాబు. ఓబులేషు, నాగరాజు, బషీరు, సంధ్యారాణి, మీరందరికీ ఓకే. సో మళ్ళా చెప్తానులేండి, ఏం చేస్తాం. That's okay. Okay, so anyhow tomorrow, okay, so I'll explain you again and then we'll see. Okay then. Okay, then I am leaving the class. Where is your attendance paper? I got an attendance paper. Okay, so I have downloaded it. So I'm leaving the class and have a good day. And tomorrow I'll explain you the same. Okay?
Okay, sir.
Okay. Okay. Good, good.